you there at home? Now okay. ಅದು දෙන්න. හරි. හරි. Online students will post a message to join. Alright. We started discussing about nucleic acid now. Alright. I'll ask few questions from that. What is the element composition of nucleic acids? Carbon, hydrogen, oxygen, nitrogen, and phosphorus also there. Alright. CHONP. So in that, what is the structural unit? Yes. What are the component of nucleotide? No, phosphate group. Alright, so this is a nucleotide. So then what is a nucleoside? Yes, a nucleotide without a pentose group. So it would, sorry, phosphate group, sorry, I'm sorry, right, without the phosphate group. So this is a nucleoside here, right? So there are two nucleic acids that we are learning in our lessons. What are the two nucleic acids? DNA and RNA. Right. So we discussed that there are two types of nitrogenous bases present in DNA. What are the two types of nitrogenous bases? Did we discuss purine bases and pyrimidine bases? So among these two, how do we distinguish these two, purine and pyrimidines? Yes. Two what? Two nitrogen containing rings are there, no? Here it is one nitrogen containing ring. Comparatively, purines are larger. This one, comparatively smaller. Okay. So what are the bases that falls in purine category? Adenine, guanine. Isn't it? Then here, thiamine, cytosine, and uracil. Go to the writing book and write this one as a table. They can question these two. So do you have a class after today? Can you inform? What time? 3:30. I'll finish sharp at 3:30. Will you be able to go if I finish at 3:30? Okay, no? Right. Sorry for keep you waiting longer. I was really like, I don't have energy today. So I did the previous class also online. I was thinking whether to get your one online or not. So I started coming, but I was late. Done? Hi. So we discussed about the base pair rule also. What is that base pair rule? Always a purine base pairs with what? Yes. With a specific word should be there. It is a must. Why is that? Can adenine go with another one other than thymine? No. So according to that, adenine pairs with thymine with 2 hydrogen bonds. Guanine pairs with cytosine with 3 hydrogen bonds. Hari neda? Hari. Write this also in the writing book. I think we wrote this in the tute, yeah. Yeah, but again, I want you to emphasize this fact again. Write it in writing book too. පුතේ online ඕකේද ඔයාට? කරගෙන යන්නලා. Tutes දෙයි පල්ලෙහි මං කියන්න මං දුන්න පහළට tutes. Hi. We are going to discuss about the structure of the DNA molecule. Hare? Structure of the DNA molecule. So children, tell me, can we see the DNA molecule to our naked eye? No, it's invisible, no? So no one knows the structure of the DNA molecule. So at the early time, the scientists started to do experiment about the structure or the features of the DNA molecule, right? So they did several experiments, and there was one experiment, we call it as X-ray crystallography. You will be sending X-rays through this DNA molecule, and the other side there is X-ray film. So there is some markings which are created on the X-ray film because of this DNA molecule. So that using that they were trying to elucidate the structure of the DNA molecule. And using this data, they develop a model that is used to describe the DNA molecule nowadays, right? The scientists who involved in this one is, we call them as James Watson and Crick, right? Those two scientists develop a model to describe the structure of DNA molecule. We call it as Watson-Crick model. All right? Let's go to the questions first, then I'll describe the structure there. Go to the question there. Asking directly to describe or asking any other sub questions? No other sub questions. Then I'll describe. Wait. So I'm going to draw the Watson-Crick structure of DNA molecule. I'll firstly tell how it formed and I'll draw that also. Wait. Now see, there is one nucleotide having nitrogenous base adenine, let's say. So here is the phosphate group. Alright? That day I explained how a phosphodiester bond form, no? Isn't it? So here, if you take this as the first carbon, second carbon, third carbon, fourth carbon, fifth carbon is over here. Here, 1, 2, 3, 4, fifth carbon is here. So a phosphodiester bond is created between third carbon attached OH group of one nucleotide and fifth carbon attached phosphate groups OH group of the adjacent nucleotide, if you can remember. So here is the phosphodiester bond. Can you remember this one? So here I'll draw a few nucleotides over here. Alright. So here, this one single structure, we can call it as what? Polynucleotide chain. Why it is called a polynucleotide? Poly means many, right? Understood? So here, then here I'm taking only the pentose sugar and the phosphate group here. This is forming the outside of the helix. That we call it as sugar and phosphate. No, it is components. That we call it as sugar phosphate backbone. Another. So, sugar phosphate backbone is there. Understood that one? I'm labeling one new thing which is relevant to 7th unit for you. Here you can see that 5th carbon is free now. So that this end of the DNA molecule, we call it as 5' end. And this side, you can see that the third carbon is free now. We call this as the 3' end. Alright? So here it is having only one polynucleotide chain. For the DNA molecule, how many polynucleotide chains are there? 2. So I'm going to draw the other polynucleotide chain. You know what? The complementary nucleotides are always pairing. So adenine is going to pair with what nucleotide? Thymine with how many hydrogen bonds? 2 hydrogen bonds. And here guanine is pairing with cytosine with 3 hydrogen bonds. Agree? Adenine is pairing with thymine with how many hydrogen bonds? 2 hydrogen bonds. Cytosine is pairing with guanine with how many hydrogen bonds? 3 hydrogen bonds. Okay? Alright? So here, the nucleotide is not visible in the same side. It's found on the other side. Likewise, the phosphate group, so pentose sugar. So you can see that there are phosphodiester bonds linking these together. Can you understand now? Hi. So here, what is this one now? Polynucleotide chain. Then what is this one? Sugar phosphate backbone. And so here, which end is free now? Third end, so that this is 3' end. Here, the fifth carbon is free now, so this is 5' end. So you can see here 5', 3', this direction, here 5', 3', the other direction. Two polynucleotide chains are parallel, but they are opposite. So we call it as anti-parallel. Understood? Alright. Yeah. So these are hydrogen bonds. We can label that too. I start drawing this one in the writing book. Oh, put it? Yeah. Pentose means 5 carbon. 5 carbon sugar. No, it's like this now. Oxygen is there. Carbon, 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 carbon. And here CH2 with the phosphate group here. So this is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon. Okay? No, no, in the writing book. Okay. At these two labels also, here it's the nucleotide. Then here it is phosphodiester bond. Shevon, have you had your exam? Oh, we have done it. We have finished. What lessons are coming? What lessons are coming? Schools will run faster, but I'm not running. There's a reason behind that. Okay, you will understand that eventually. I'm also going faster, but it doesn't mean like I'll be running without doing questions, right? Questions are the most important thing. Most schools don't do questions usually, right? And even the classes also. Are you done? Right, this diagram is for your understanding. If you are asked to draw double helical structure of the DNA molecule, this is not what you need to draw. Finish labeling and tell me, I'll erase this and I'll draw the correct diagram. You need to draw it in the examination. You're still doing, no? Do. Don't worry. Doing? Okay. Right. So, shall I erase then? Are you all done? This is question in the new syllabus 2021 paper to describe the double helical structure of the DNA. This line with a thickness, we call it as sugar phosphate backbone. And this is one sugar phosphate backbone. And here's another sugar phosphate backbone. And so you can see that sugar phosphate backbones are found in the exterior of the helix and the nitrogenous bases are paired in the interior of the helix. So we will draw a few nitrogenous bases also here. Adenine plus thymine with 2 hydrogen bonds, guanine plus cytosine with 3 hydrogen bonds, cytosine, guanine, thymine, adenine, adenine, thymine. There's no particular order. These nitrogenous bases can arrange in different way. This one might be having different nucleotide sequence. In myself, having different nucleotide sequence. You are having different nucleotide sequence, right? So we all are having unique sequence of DNA nucleotides, right? Right? So you have to label these things there. Mm. In the examination, they took this complete thingy, DNA double helix. Here nitrogenous base pairs. This is sugar phosphate backbone. Right? Draw this. This is the diagram that you need to draw it in the examination. This is the double helix structure what we drew to understand before. So that is not the double helical structure. Why do we call it as double helix? This is having two sugar phosphate backbone and it is twisted around an imaginary axis. Right? Yeah, I'll tell you that also in a little while. Just draw this one, Sona. Hmm? Yeah, so from what we discussed, yes, true. Hello. Listen, will you now? Okay, draw and finish with it. And I told you, no, there will be an exam coming up. Week after, 1 week we'll do the theory. Right? No. Uh, I'll confirm about it because 2 weeks I'll take to cover the practicals also. No, there are two practicals, first and second. Uh, I'll give you the date today. Wait. We'll have the exam on 28. Not imaginary axis, interior of the helix. It's now, like this is the imaginary axis. Around that only, this is folding. We can't say exactly the hydrogen bonds are there inside that in the imaginary axis. No, the whole helix is turning around the imaginary axis, right? No, around the axis, the bonds are found, right? For example, I'll show you a model. Like this, turning around the axis like this. Okay, got it? You're okay? Yeah. Right, so here you can see this one. This is how that it really coils. Look at this one, Putisheva. So here we have a big turn and big turn here and a small turn here. So usually you are not learning these names. I'm just telling. This we call it as major groove. This one we call it as minor groove. So major groove and minor groove together we call it as one complete turn of the DNA molecule. This one complete turn consists of 10 base pairs. Understood? 10 base pairs are there in one single turn of a DNA molecule. Understood? Draw this too. This one you don't need to memorize, just for your information. Oh, that's what I was about to decide. Right, even if the content finish, I'll be continuing the lessons on 14th and 21. 28th, you'll be getting the exam, including the 2 practicals and up to nucleic acids. That means nucleic acids also included. 21st, 20 issue this. Then we'll have on 21st, if you don't mind. 21st, come on. Harena, you need to do me a favor. I'll be giving the tube today. The question tube. You have to finish the possible questions that you can do and come to the class. Then we'll discuss and finish. I don't have time to give it to you to give the time to do it at the class itself. Right? Do it and finish it and come. Then we'll discuss sooner and we'll discuss the practicals too. Let's go. Right. Now, even if you want to have one turn, you have to complete this. One turn, 2 together. Right? Yeah. No, don't mark like no. Don't mark like that. Just draw it simply and mark. Alright, go to the question that I have asked to describe the Watson-Crick model and write this exam question also. This is 2021 A level. Question number 5, part B. Describe the structure of DNA molecule according to Watson and Crick model. Right, my question is asking to describe the structure of the DNA molecule. Is that so? My question? Yeah, same question. Eh? Is it mentioning Watson and Crick model? Then it's okay. No? So if discussion can come in a different way also to describe, asking to describe the structure of the DNA molecule. That way also the question can come. In that case, you have to add one more sentence telling that it's described by the Watson and Crick model, right? Okay, double helix structure of the DNA molecule. Let's go and write the answer there. We are writing it point form. Number 1, DNA molecule consists of, yeah, writing in the table. Molecule consists of 2 polynucleotide chains. Which are spirally arranged around an imaginary axis. Around an imaginary axis. And forming a double helix. Number 5, sugar phosphate backbones run in opposite directions. Number 6, and it is called antiparallel. Number 7, sugar phosphate backbones run in opposite directions. Number 8, and is called antiparallel. That one, wait, I'm repeating the same. Sorry. Number 7 is not this. Number 7. Sugar phosphate backbones are on the outer side of the helix. Sugar phosphates Number 8. Nitrogenous bases are paired and are interior of the helix. Correct? Number 10. Two strands are held by hydrogen bonds. Number 11. Between two complementary nitrogenous bases. Number 12. Adenine pairs with thymine. Number 13. Thymine pairs with guanine. Correct? Cytosine, yes. Right. Number 14. Two hydrogen bonds between adenine and thymine. Number 15. Three hydrogen bonds between guanine and cytosine. Number 16. Two strands are complementary. Complementary to each other. Number 17th, in a one complete turn of DNA, there are 10 nitrogenous base pairs. Correct? Correct. Right. Now we'll give you 10 minutes time to study this now itself and finish. Whenever you are asked to write the structure, a diagram is necessary. Since we drew it now, I'm not asking you to draw it again. So please make sure that you draw if it is asking a question paper, right? Alright? Alright, study. Finish? Right. Let's go to the next questions also. Right. Asking the functions of DNA. What do you think? What are the functions of DNA? They are storing genetic information in a sequence of nitrogenous bases. So many information is stored there. You know, like certain characteristic features of us is stored as genes in DNAs, right? So genes are certain nucleotide sequences found in a DNA molecule. Right? Right. Go to the book and get the functions there. Get the functions of DNA as it is given in the book. Have they given? Yeah. Not only storing, what about transmitting the genetic information to the next generation? It's also done by DNA, no? Isn't it? One thing is storing, one thing is transferring. Yeah. That means, yeah, let me explain that one minute. Wait. So sometimes people are telling that your hair looks like your mother's hair, your eyes look like your father's eyes. Likewise, so the information which are stored in mother and father, those genes are coming to you. How they are coming to you? They are gametes. Egg is bringing from mother, sperm is bringing from father. So these genes are now given to you. So at your body, genes are expressed. What's the meaning of expression? Gene is making a protein, right? So if protein keratin made in your body, it's making your hair curly. That means the information which are there written in the nitrogenous bases in the DNA is now expressed, right? So DNA is storing genetic information, transmitting the genetic information, as well as it is giving the information to synthesize proteins. Understood now? Right? Study and write that. Now we are going to talk about different types of RNAs. So there are three types of RNAs in the body. Now look at the board, everyone. There are three types of RNA. Number one, mRNA. What is the long term? Messenger RNA. Correct? Number two, tRNA. Transfer RNA. Number three, rRNA. That is ribosomal RNA. Got it? I'm telling what are these three. Think this is the nucleus. In the nucleus, DNA is stored. So DNA is found as the network of chromatin over here with the nucleolus here. Alright? So I told that nucleotide sequences in the DNA molecules are acting as genes. So think now, here it's one gene, here it's another gene. Likewise, numerous genes are there on the DNA molecule. So let's say you want to express one of these genes, you are making a mRNA molecule according to the information which are there in the DNA. That means it's copying the information which are there in the DNA into a ribonucleotide sequence, right? And this is now coming to the out of the nucleus to the nuclear pores. And it is coming to the site of protein synthesis, which we call it as the ribosome. So here is now mRNA. Alright? This ribosome is made up of rRNA, right? Correct? Large and small. Large and small one, both are made by the rRNA, which is made from this nucleus. Those are also produced inside the nucleus and now they are in the cytoplasm. Or else ribosomes are found in other places such as in rough endoplasmic reticulum and the chloroplast and mitochondria. Those places also we can see. And now this information is little by little they are read by this ribosome. According to that information which are there in mRNA, tRNA molecules in the cytoplasm they are coming with relevant amino acids. Alright? So this is tRNA. tRNA is kind of a linear molecule, but it's folded. There are complementary hydrogen bonds in these locations, right? So this tRNA molecule has a characteristic shape. We call it as clover leaf structure. Why is that? It looks like a clover leaf like this. Okay. So that's why we call it as clover leaf structure. But in some books they say it as hairpin structure. You are not learning that, but the hairpin structure also is a very famous term among the molecular biologists, right? Hairpin structure. You know that black color hairpin that you use, small one that having clip like this. Okay, hairpin structure. Alright. So this one's function is to bring the relevant amino acids to the site of protein synthesis, which is the ribosome. So let's take one by one the functions of these RNAs. mRNA, what does it do? Copying the information in the DNA molecule and bringing it to the site of protein synthesis, which is the ribosome, right? They are through the nuclear pores. And then the rRNA is making the site of protein synthesis, which is the ribosome. And then tRNA is bringing the relevant amino acids to the site of protein synthesis. Understood now? This is well explained in your 7th unit. I'm just giving the basic idea what's the function of all these. Haridhar, right. Go to the question now and try the 3 types of RNAs. What's the next question, Putte? mRNA is a linear molecule, no, isn't it? What else we can say about mRNA? What else we can say other than saying linear? It's the least abundant mRNA, no? Structure we can't say that, but we can talk about it, right? Why it is having like it is least abundant on the in the male cell? Yeah, that's what. Once if the protein is, sorry, if the DNA is transcribed only, that means if you need to make a protein only, it's made, right? Otherwise, no need. But still, tRNA and rRNA, they are found abundantly in the cytoplasm or cytosol, right? Hi, let's write that answer. Wait one second. That's linear molecules. If the structure is asked, that's only you need to write there. Then what's asking the functions now? Functions now. Next question. As it is, you have to write the functions given there. Copies the genetic information stored in DNA molecule as a sequence of nitrogenous bases for number one. Number two, transports genetic information from nuclear plasm. Nuclear plasm is inside the nucleus, right? To the site of protein synthesis is synthesis through nuclear pores. Okay, if you want, you can draw the diagram at somewhere else. Describe, ask it to describe a structure. To describe the structure, right? Nature. Then you can write it is a linear molecule and it is the least abundant type of mRNA, type of RNA here, right? Okay. Alright. Right. What's the next question, Putte? Let's go to that one. So what can we say about it? It's linear, but it's folded due to complementary hydrogen bonding. And what's the name for the structure there? Clover leaf shape, right? Let's see what's given there in the book also. Book says that smallest RNA molecule, linear, but forms three-loop structure as shown in the diagram. I write that and add these two names also within brackets. You will need this also if you are referring the books, right? Right. Let's go to the functions of tRNA. What's the function? It's bringing amino acids to the site of protein synthesis. Did you write that? Write that function there. Transportation of amino acids to the site of protein synthesis. Right. Make that question into tRNA and write the function there. Mm, right, let's go to the rRNA. rRNAs are having irregular shape. No, other two are linear, but this one is having an irregular shape. So rRNA has a complex irregular structure. It is the most abundant type of RNA. It provides a site where the polypeptide chain is assembled. What is that? What's the meaning of that? In this place only, the polypeptide chain is formed. Amino acids are linked together and it's forming a protein. Right? Okay? Another. So we discussed about four levels of protein structure now. Among that, which level is synthesized here? Yes, primary structure. I told that it's not functional. We don't call it as a protein even. Thereafter, correct folding should be there to have the functional protein. Understood now? Hard. There's one question missing neither. Write that question there. Describe the nature of the rRNA and the functions, right? Two questions you have to add. What's the next question, Putik? Or what's the next question? So you have to say that the most abundant is rRNA is the highest, then tRNA, Then the least abundant is mRNA, no? Isn't it? I'll write that there. Alright. Let's go to the next question. What is asking there? Right. Okay. I'm going to explain that. The other nucleotides other than these ribonucleotides and deoxyribonucleotides. Hmm. ATP is one nucleotide. What is the long name for that one? Adenosine triphosphate. So what is this one? Our energy molecule, no? So this is providing us energy. So this is important. And we will be learning about NAD plus and NADP plus and FAD plus name okay FAD with right no plus right FAD those are nucleotides which are important in our certain reactions. NAD plus and FAD are found in cellular respiration lesson, right? NADP plus is found in our photosynthesis lesson. Okay, children? So, these are important. These are usually acting as electron carriers as well as since these molecules can reduce by taking hydrogen, let's say NAD plus can take hydrogen and reduce into NADH, they can reduce themselves while oxidizing something else. Alright? So they can act as oxidizing agents. Understood? So these are electron carriers and oxidizing agents. Alright? Right. Come to the book that they have given that nicely. ATP is the universal energy carrier. NAD plus acting as a coenzyme. What is a coenzyme? For certain enzyme activity, non-proteinous components are needed. That may be organic or inorganic. Coenzymes are organic cofactors, right? Okay. You'll be learning in coming up lesson this is properly. And then acting as function as electron carrier, function as oxidizing agent during respiration. NADP plus act as coenzymes acting as electron carriers. NAD plus act as an oxidizing agent in the photosynthesis. FAD act as coenzyme, act as electron carrier and as well as acting as oxidizing agent. Harry, study this now itself and write this down. You will be familiar with these molecules well in the coming up lessons, right? And these should be correctly written there in the exam papers. You are not giving marks for NADP instead of NADP plus. FAD only, no FAD plus, right? Write correctly. Hmm? It means us, we have ATP as the energy molecule and bacteria, almost all the organisms are having ATP as the energy molecule, okay? Sure, sure. Wait, wait. Now think NADP plus is there. No? It can take hydrogen. If something is taking hydrogen, it means that it is reduced, right? So this is forming NADPH. So this substance is reduced. While it is reducing themselves, they can oxidize something, as you have heard about redox reactions. If something is reduced, simultaneously something is oxidized. So this is reducing itself while giving power to oxidize some other substance. Understood? Another. So these all can act as oxidizing agents. If you take it the other way around, NADPH can turn into NADP plus. It is oxidation. NADPH has the ability to act as a reducing agent. Okay. All done? Did you write the answer there? Alright. There's an exam question also relevant to this area. A little before that, I'll give you that to write at home and come. 21 A level, the first part of the question that we did discuss today has come from this section, or little early nucleotides. Let me see. I write down this question. Describe the components of, this is 2021, don't look at the answer and write. Yeah, question number 5, part A. This is the first part of the previous question of the DNA molecule, right? Describe the components of, this is not a direct question that we wrote before. You have to think a little and you have to understand what to get to the question. Let's see after you are done what is necessary to include. Describe the components of nucleotide, let's say, nucleotides, and explain how nucleotides form the backbone of DNA. You need to include the diagram also. Don't forget that, right? The label diagram of a nucleotide should be there. Alright? Are we having any other questions in this section? Alright, this is a good question. You can take a little time and write the answer there. If you are not sure, use the pencil to write the answer. Alright, quickly write. We'll give you 2-3 minutes. If you are getting late, you can go. Only you having class, ne? Both of you are here. Don't worry, sir will finish later. Today there are some holiday, ne? Eid. Today? Yeah. No, Bhute, I'll mark and give this week. I was really busy with the school examination papers. Sorry for the delay. Lipid and protein is with me. I'll give it to you sooner. Your one, I didn't mark any essays, neither. Hurry, I'll mark and give, okay? Sorry, others I gave to the others. I couldn't give proteins and lipids to you guys, right? As soon as I mark, I'll send you a photo also and I'll bring it to the class also, okay? Hi. Right. What are the differences? What are the, sorry, wait. Right. What are the structural differences between DNA and RNA? We'll write the best ones first. Apute? DNA is made up of what? Deoxyribonucleotides. That's the best difference between these two, isn't it? Then what about the other one? Made up of what? Ribonucleotides. Huh? What else? No, that is the difference between no ribose sugar and deoxyribose sugar. We are not writing that. What else? There's a big difference. This is a double-stranded molecule. This is a single-stranded molecule. Okay. Right. Then anything else? Structural differences. Right. Let's go to the chemical differences then. Chemical differences. DNA has adenine, guanine, thymine, cytosine, nitrogenous bases. Instead of that, here RNA, what is there, Pute? Adenine, guanine, adenine, there's no thymine, uracil, and cytosine nitrogenous bases present. So this can be written in another way also. How do we write that? Thymine is present. Thymine is absent, no, isn't it? Correct? Here, what else we can write? Uracil is absent. Here, uracil is present. Any other chemical differences? Here, deoxyribose sugar is present. Here, ribose sugar is present. Yeah, good question. I guess in this 2021 A-level paper, I think, no, they took signs, symbols they took, but if you have mentioned it without telling that having adenine and thymine having two nitrogenous, two hydrogen bonds in between, they have not given marks for this one, right? Adenine and thymine, guanine and cytosine. Likewise, if you have written, you are not getting marks. You have to write that adenine and thymine makes two hydrogen bonds in between, and guanine and cytosine producing three hydrogen bonds between. But these are standard ones you can use, no issue, right? Write and finish. Right. So now DNA is making complementary base pairs. Now what about RNA? Is it forming complementary base pairs? Yes, there's a sentence in the book that's telling that tRNAs are making, and also another RNAs can make with another RNA also. There's one statement. Let me see what it is. Open the book. Hmm. Here in the structure of RNA section, second sentence, complementary base pairing between two RNA molecules or within the same molecule may occur in some. Please highlight it and keep. It can come in MCQ sentences. Hurry. Putte, don't forget to collect the tutes from the downstairs. Uh, Sometimes they may tell that to pay the month fee and get that one. If they are so, get the question tute only. If you have not paid yet, tell them that I asked to give the question tute only, right? If you have not done the payment, right? Okay, then we'll see you on next day. Sorry for the delay today. Others online students are not here. I think it's because I got late. One is here, right? Then we'll see you on next day. Okay, study well. Now you have done a big section. Okay, the biomolecules you have finished already, and there are some questions in this tute as well. Please try them and come. Okay. See you. You can go. Bye. I put in. But it was in my essay, I dropped the points, so that's why I took out of 150. Usually it gives out of like 4 marks into each point. Good one. Haripute, see you. Still, I couldn't mark the papers, so they gave me the